Professor Vanderlei, just a short introduction, is a titan of the Brazilian physics. He graduated in physics and material engineering at University of São Paulo and Federal University of São Carlos. And later on, he moved to Massachusetts, where he got his PhD in physics degree from MIT. He's currently a full professor at the University of São Paulo and has been the coordinator of the USP, University of São Paulo Agency of Innovation. He works in the area of atomic physics and applications in optics and health science, such as cold atoms, bosons and condensates, and photodynamic actions in cancer and microbiological control. And in this multidisciplinary field, Bagnato has achieved a rather long publication record with over 500 articles published, along with 24 book chapters and several books. So his research, of course, led to the formation of many students, over 45 master theses and 36 PhD degrees were advised by him in areas in physics, dentistry, and medicine. Professor Bagnato is also a member of the Brazilian Academy of Science, the Academy of Science for the Developing World, and the Pontifical Academy of Science of the Vatican, which I think is a rather unique position there, and also the National Academy of Science of the United States. So with no longer ado, welcome Professor Bagnato. The stage is yours, Professor.
Well, thank you, Gustavo. Let me put up my presentation. I hope, is that on?
Yes, Professor, we see it.
Okay, so I will put in the mode of a presentation so we can all see. I'm gonna, well, first, thanks the organizing for let me say a few words about this work. And that will be a little different from the last few talks. You know, even though magnificent talks we had this morning, I will speak a little bit more on the other side, which is the application of light to interaction with the life science. And I will focus in something that's very on fashion now, that's microbiological control. Okay. You see, first, before I go on, I would like to communicate that today we lost our founder, the founder of our institute, Professor Sergio Mascarenhas, passed away this last night, and I will dedicate this talk to his memory. He was a very important Brazilian scientist, worldwide recognition in many initiatives, and we lost him at the age of 93. And so I will, after my talk, go to the funeral, and but I would like to dedicate in his memory this presentation, which is nothing compared to everything he has done. I would like to start by saying that actually we've been very modest in this community. You know, we never recognize how important is the photonics nowadays. In fact, on the 19th century, we had the century of mechanics, you know, big machines and everything, motors and everything. In the 20th century, we had the century of electronics. But now there is no question that we are getting to the century of photonics. And photonics is being really, really important in almost everything. Imagine communication, which today is the key word for the whole civilization. We could not do in the way we do without photonics. So actually, we can claim that we really are getting into the century of photonics. Photons will be everything. Instead of using electrons for many things, we are already using photons for many things. And now we're going to start a whole new area of quantum communication. Some people believe that we are on the quantum area, but that's not true. We're still going to use light for everything, for preparing, for reading, for everything on this new kind of domain. Well, I am in São Carlos, and I have a big team of people working in different things. And we work in atomic physics with cold atoms mainly, but some experiment with non-cold atoms. We work in biophotonics, that will be the main topic of what I'm going to tell you, and nanoplasmonic, which is the interaction of light with nano metallic structures. And in a second kind of level, we do a lot of science diffusion, innovation, and especially innovation with social responsibility. I'm not gonna keep putting my publications on this, but all the publication of my group you can find in your homepage. And I'm not gonna give all the names as well because it's huge. We have more than 140 people working in your group today, or in your center, I should say, with many, many different laboratories in many fronts. Of course, everybody that has white vest, they work with some clinical stuff or some bio stuff. We do a lot of diffusion, and this is important because people has the right to know what are the scientific advances coming. And if we educate well the youngs, we're gonna have a lot of motivate people for this field. So we do a lot of exhibitions. We create new mini laboratories in optics and many things for the young students. And this is important because we have to, everybody has to keep doing diffusion of science because we needed to show how science is the key thing for everything on the advance of society. Okay, well, on the atomic physics, I do a lot of atomic physics with cold atoms. We have many laboratories. I'm not the only one. We have more people doing that. We have Marcassa, Felipe, Sergio, Muniz. We have Patricia. We have Emmanuel. They have different laboratories doing different things. And recently I've been focusing study the disorder in the quantum world by studying turbulence in quantum fluids. But this is not, oh, and by the way, we have the only and first atomic clock built up in the South Hemisphere. It's a atomic clock based on cold atoms, and is considered as the most prominent new standard for time and frequency, right? And this is very interesting because precision is part of very good telecommunication as well. But I'm gonna talk about the biophotonics. And basically what we do in our lab, we try to use light to interfere in many different ways with the living objects. And we especially use the photodynamic therapy for cancer, for microbiological control, and most recently for food safety and agriculture. And we normally go from principles, which means do experiments just to advance the knowledge, until clinical applications. And we are a physics institute with a bio, reservoir of animals here. I forgot the word in English for this, I used to know. Anyway, but we do take as a group, as a center, some challenges, and our challenges are in microbiological control, even before this pandemic situation. We know that the resistance to antibiotics bacteria is a big problem. They are announced to be the next big thing, especially now that we spend 1 year really using a lot of antibiotics. Now we have a lot of bacteria that are resistant to antibiotics. And people are anticipating a new pandemic situation with those diseases. Cancer is also a big problem because if we don't die of anything, we're gonna die of cancer. And chronic diseases also is a big problem. And food safety, which I don't think I'm gonna have time to talk about, but it is a very important issue because especially for Brazil, where we export 25% of all the grains produced in the world, or consumed, I'm sorry, in the world. And we are big producers of meat and many other type of food. And we have to worry about how is that being done. And we know the scandals we have recently on that issue. So we work with that. And normally we use as principles light. That's why I am in a community of photonics. And we use the photodynamic action, which is basically a photoreaction, producing free radicals that can oxidize either microorganisms or cells that we want to destroy. But we've been using also ultraviolet, because ultraviolet is very good for acting on microorganisms, especially the virus, because it destroys the genetic material. And we use also light for manipulating metabolic reactions, and this is what they call photobiomodulation. But I would like to show you some of the infections that are being treated basically with photonic techniques. Light is one of the most important instruments in this sense. And all this that you can see in this diagram is being somehow treated with photonic techniques, right? And we are kind of pioneer in a few of them, including the periodontic disease, including the onychomycosis, which is a fungus of the nails, including the condyloma, which is the HPV, and also the respiratory tract, to trying to treat the pneumonia by extra body illumination, as I'm going to show you. Normally, the technique involves putting a molecule, whatever you want to treat. And of course, we have many work on the basis of all of this, but I'm going to show you kind of highlights of things. And this is the treatment of HPV, which is the condyloma. It is a virus lesion, and it is the main cause of cervical cancer in a woman. And you need to treat the lesions because in place, they claim that we have more than 20% of all the women in the world contaminated with HPV. Here in Brazil, we know this number, and people are vaccinating young people to prevent them to get this, but still far from control. And we have to fight the lesions. There are many problems in doing surgery. You're never going to cut a virus lesion like this because you're going to spread everywhere. But there are cryo type of procedure, but the most convenient is the use of photonic techniques. A cream that's put on the lesion and illumination. I know that the HPV virus is on the body, systemic, but the lesions are the main focus of reproduction and transmission. And we've been treated that with great success for humans, and we are part of the new technology for human health in Brazilian program, and this is going very well. On the onychomycosis, it is also a fungus that's very hard to kill because it's on the knit of the nail, and the way to do that is using a cream that creates pores on the nail, put another substance that here is on the base of curcumin, is a curcuminoid, right? It's a molecule like the one you see, and that absorbs very much light, converting the oxygen in singlet oxygen, converts the triplet in singlet oxygen, resolving the problem after a few applications. And that's very important because the consumption of fungicide is giving a problem for liver. In general, normal people are consuming more fungicide than they should. And then being able to attack the disease with the photonic techniques may be a way to not having complications for liver cirrhosis and things like this. But that's not only the things we do. We also developing techniques for cleaning organs for transplantation. So suppose you are a donor of an organ, so hope not soon, but if you are, and then you have some disease, they cannot use that organ. But we are in a demand of organs. You know, there is a meeting that happens a few months ago in the Vatican Academy of Science, and they claim that only 10% of the necessary organs for helping people to survive to diseases is being available. And sometimes they are just discharged because of disease. We are developing techniques that allow with light to clean it up, the organ during the perfusion. When you have an organ, you have to circulate a liquid for preservation. And during this procedure, we are removing microorganisms from the organ. And then we clean it up. The idea is clean it up, this fluid, and then you are cleaning, you are like washing out the organ. Why not use 100 liters of this solution? Because it costs $2,000 a liter. Now, some virus or some bacteria still are retained in the organ, and that comes the photodynamic, because we also add the photodynamic to the procedure. And as you can see in the plot, we decrease quite well the virus charge of the organ as well as the infection. We are doing lungs in connection with Canada group, but we are doing kidney and liver here in Brazil, and hopefully we'll be having this very soon. But now we are in the respiratory time of disease. We know that we are suffering. We are in a pandemic of this. And then I'm gonna show to you a selection of work we have been done for the last 10 years to somehow clean it up the respiratory tract, starting from the oral cavity, going to the nose, larynx, trachea, bronch, and lungs. Normally, to do this mouth decontamination, you just need to wash out, to rinse the mouth with some solutions that are harmless for you until I put light. And then you rinse the mouth, the bacteria absorb the light, I illuminate by 5 minutes, the whole operation takes 10 minutes, and turns out that you decrease quite well the amount of contaminants in the mouth. This is good for people that are in contact with contaminated people, because when you are in contact with, for instance, a people that has COVID, you may get the virus, but the virus is staying in the nose and in the mouth for a period of time. This is called the residence of the virus in the entrance. And then by cleaning up, we may save a lot of people. And of course, after a while, there will be a recovering of the population of the microorganism in the mouth. And this work has been done long ago with many, many participants, including the wife of Kleber Mendonça, that was my student long ago. But we want to treat things that are contributing to development of bacterial resistance. And then pharyngotonsillitis, which is the throat infection, is one of the most important. And the idea, this is a big problem. Very, very big in the whole world. It's very big problem. Everybody experienced many, many antibiotics in the early stage of their life. And now we are getting, even as old people, a lot of necessity for antibiotics because we are contracting a lot of throat infection. Okay, so the idea here is very simple. You give a kind of a kind of candy for the people to swallow, and they stay on the mouth for about 3 minutes. Is based on hydrogel and curcuminoids molecules, and that dissolves and goes to the plaques on the throat, and then they get impregnated there. And then after illumination, we're gonna kill the infection. This is being done in many, many situations. We have a kind of very nice collection of work on that. And this simple result, simple technique, you can do almost everywhere because it doesn't require anything. And the number of bacteria and the infection comes down many logarithm orders. And we have done this. I'm not going to talk about the devices because they use fluorescence to detect the infection and to see if the procedure was well carried out. At the moment, we are doing many groups, and this is the way to deliver the substance, is by having a lollipop or a candy or even a syrup that you take, put in your mouth, and swallow that slowly, and that is going to the plaques, bacterial plaques, And the result is being wonderful. We've been getting on adults 92% of elimination of the infections without the use of antibiotics. And in youngest, in kids, we've been able to really reverse the infection picture in 24 hours. Of course, we are now in a clinical trial 3, which means being assisted by the authorities to really implement this technique of pharyngotonsilate disinfection. And this is great because we can get even elimination of virals, and I don't have time enough to go to all of this, but we can accelerate even the action of antibiotics when we use it all together. I'm sorry, I went too fast for that. But there is some also problems presently that needs the control of infection. And one of them is the intubation. When you get this endotracheal tube, and this is common now, right? People don't hear every day in the news of the TV, intubation, intubation. We are intubating everybody. And when you do that, this strange object in your trachea is a cause of infection, accumulates microorganisms, and then you get pneumonia. Getting pneumonia, you may die if the pneumonia does not respond to antibiotic. So what we have done with photonic techniques is a way to prevent the infection to take place when a patient is intubated. So how we do that? We take the tube of intubation, we functionalize it with a molecule. So we stick to the walls, a molecule, and we're gonna have an optical fiber that goes inside the tube together with the intubation, which is going to illuminate. And by illuminating, we prevent when the bacteria, because the surface of the tube is the main region for multiplication of the bacteria and the colony's adhesion. By illumination, we prevent that to happen. So here you see how we have done the development of the infection and what's happened when we just illuminate the tube from inside with a lower power. We are talking about 100 joules per square centimeter, right? And then we prevent the infection to take place. That's how the tube looks like. And that's the two plots. One is the progression of the infection or the contamination when we do nothing. And the other one is when you are illuminating. When you are illuminating, basically we are suppressing the formation of infection. Now to keep going with the work, the student here, and together with a very nice postdoc, Katie Blanco and Amanda, they produce a very nice artificial trachea for the whole investigation before we going to the humans. And I hope this will happen very soon. It's very safe because the illumination activate only molecules that are sticky to the surface, so does not hurt the trachea or anything else. So that's a very important advance if we can succeed in implementing this in clinical use. But this is even more important, is the treatment of pneumonia. Suppose you got the infection, how we treat that? Okay, the idea then is very simple. And this is the device we built it for doing the animal, small mice. And now we're going to pigs. And the idea is easy. We suppose you have a pneumonia, we give a drug by inhalation. So you breathe the drug. And then we illuminate by external illuminators. And this light in the infrared is enough to penetrate on the thorax chamber, hitting the colonies that are already with the drug that we inhalate. And then we start to kill the bacteria without really invasive techniques and no antibiotics, because most of those infections, they are becoming resistant to antibiotics. This is a diagram that shows the survival. And the black one are the little mice that we didn't do anything. They die as the time passes. And the other one, the red, are the ones that we treated, and none of them die. So means that we are able to really kill the infection by this procedure. Well, now we're doing many, many different experiments because we want to get to the real human infections. We did the machine, but first we have to do in pigs, and that's why we're doing at the moment. We are treating the pneumonia that was induced in pigs. And of course, we are doing very well. We are understanding the problem and understanding the challenges to overcome. But we believe very much that this will be a great success, and we'll be able to eliminate basically the pneumonia, the infection of the lungs, by doing breathing and extra body illumination. In the pig models, we have the seed microorganism inside the real pig thorax, and then we see the elimination as of the infection as we illuminate. In the real life, we have some problems, and then we are trying to solve the problems, including looking to the diffusion of light. I know this. Thank you. I know 5 minutes left. Okay, I will speed it up. And then the thorax is very nice because it's a cavity. The diffusion light produces a uniform illumination. So we create a very nice system for killing. We have a participation of many people in this, even with international collaboration. Well, I could not forget to say our work on cancer. We're doing many different types of cancer, and skin cancer is one of the most important target we have, using the photonic techniques and even detection by fluorescence and a sensibilization with porphyrin or induced porphyrin drugs. And we've been well succeed in doing the skin cancer treatment. In Brazil, skin cancer is a huge problem with more than 250,000 new cases every year. And we have spread many centers in Brazil with this program using photonics. And now we are making the treatment and already reached 95% elimination of the lesions of the skin cancer, and now improving with even more technology like the dissolvable needles. And I only put this because we were the cover of Physics World with this work. Also the cervical cancer and also the elimination. And just to end up, of course, I spoke a lot about resistant bacteria to antibiotic. Can we use photons to bring back the bacteria to be attacked by the antibiotics? And the answer is yes. We can take resistant bacteria and give a shot of photodynamic action, and by oxidation, they may lose a little bit of the resistance. And then the antibiotic became again useful and a very nice controlled way. I told you in the very beginning that we are also focusing in elimination of contamination in foods, and we're using equivalent techniques, combination with UV and photodynamic action as well. And this is being very well succeed. And in fact, in the agriculture, we can think about using the sun, which is the most important photonic source we have, a lot of photons. And now we are thinking to do many, many applications in the agriculture by spraying some substance that the sun will activate and eliminate the drugs or the bugs or whatever is the disease that the plants may have. I was very fast. I give you a tour on how photonics is important for life science. We saw many talks on the first day on that topic, wonderful talks. And but little of them really touch the microorganism or infection problem. So, and since this is the limitation we are facing now and we're gonna face it for the next coming future, I think photonic will make a revolution in the microorganism control. Well, I will stop here. I still had more to say, but I see the chair of the section became very upset with me. And I have this principle that people say that the audience may forget what you say, may forget what you show, but never will forget how you make them feel. So I hope, I don't want to make everybody tired, and I don't want to make everybody irritated, because I want all the audience to remember me forever. Thank you very much.
Thanks, Professor Vanderlei. That was a fantastic talk. We already have a couple questions here. The first one was from Michelle. So Michelle asked that he couldn't understand how the disinfection mechanism occurs for the intubation apparatus. Could you tell us again how the light interacts with the chemical substance and the bacteria, please?
Yeah, okay. First of all, the chemical is sticked on the surface of the plastic tube. But the bacteria gets there, and there is oxygen in that. We excite the molecule, we excite it to a triplet state of molecule. I didn't go in detail. And then when the oxygen collides with the molecule, it gets the energy, and the triplet oxygen converts into singlet oxygen. That is a very oxidative agent. And then in nothing, it oxidizes everything around. And what's around? The bacteria. So we kill the bacteria by photonic oxidation. And it's very efficient. With very little light, we can eliminate infection very, very efficient. So this is a very good hope. We already demonstrate all the principles. And we know these principles of elimination of bacteria for a long time. Now we are kind of incorporating in special devices that normally are the cause of infection in some situations.
Okay, there's another question, comment by Professor Nilsson. He says, very nice talk. Did your group develop any photonic use for killing the coronavirus?
Yeah, you know, UV is normally the radiation that people uses to kill the virus. But we have been doing many virus study on the hepatitis for cleaning up organ transplantation. And the photodynamic action is also very efficient for that case. And not only our group, but the world is developing ways to protect the people by cleaning the nose and the mouth. It happens that we are demonstrating that long ago. And normally all the techniques that kills well using photodynamic action, bacteria, gram-negative bacteria, they kill virus much easier because the virus do not have all that protection. It's just the genetic material and a proteic capsule. And they all destroyed by photodynamic action, by oxidation. So we are doing, yes, and but the world is doing too. And of course, but we have to do because this is becoming more and more important.
Okay. There's one last question that I'd like to bring up. Everybody is making excellent comments about your talk, but also there's this important question from Alexandre Paul, Paraná, that he asks, what are the difficulties that your group is facing to bring these treatments into practical use? I think that's very relevant.
Well, you know, you always have to follow a sequence of events to bring any new technology to the people. Like skin cancer now is a reality in Brazil. You can go to a dermatologist. They may offer the technique that we help to develop it, and even the equipment. You may, very soon, you're gonna get a disinfection of your throat with our technique. Those things are being approved to clinical trials, and of course will be approved by Anvisa as techniques. The skin cancer is being close to being incorporated in the SUS, in the health unit system in Brazil. We are very close to be accepted as one of the techniques because it is cost less than the conventional surgery and less complication. But for the elimination of the pneumonia, we hope to get to clinical this year, but we have to pass through the pig experiments. And then, of course, it's always, you face a challenge every day, and we are about to overcome one of the challenges we have to make efficient activation of the drug inside the lungs. but all the illumination device and everything is being developed, and I hope this will be soon. The decontamination of organs for transplantation is already on the hands of a few companies, a few startups and a few well-established companies. They're gonna adopt this as a way to clean up organs for transplantation, which I hope will increase the availability of organs for people that need.
Okay. Thank you very much, Professor Vanderlei, for your insights in this.